Okay, well, thank you for coming back. Appreciate your patience with everything. This is E340, 542, Lecture 3. I need to remind you the class is recorded and live streamed. Today, we're going to start working on SIR models of epidemic, which are the classic simplest models that people use of population epidemics. So we're going to start out by having students present some of the papers that they found quickly, and then we will do a few reminders on the SI models that we started on last week, and then we'll do some exercises both in terms of plotting and in terms of understanding SI models and then SIR models. I hope we'll get to some of the simple analytical results that you can derive from SIR models. And depending on how far we get today, we're going to talk a little bit about how to estimate parameter values in SIR models. The main homework for next week will be presenting your ideas for a project. I realize that we're pushing that really early this year. The idea is to have some preliminary ideas that we can work on together to try to make sure that everyone has a good project, especially if people want to team up. I wanted to make a few comments on the homework. There were a few people who didn't submit homeworks. I hope that they were okay. I know one person was a little bit thrown off because the Canvas assignments are not always perfect. Canvas has a tendency to lose equations and figures that are embedded in homework texts. And so in general, I will give you by email and then also upload both a Word version of the homework and a PDF of the homework assignment. People found some interesting papers and some interesting ideas for networks. One thing is whenever you refer to a paper, you should always automatically give the citation for the paper. So several people wrote paragraphs saying they found an interesting paper and then describing the paper's contents, but they didn't say who wrote the paper or what it was. So you should always give the citation whenever you discuss any paper. If you don't do that, it's not so easy to figure out what's going on. If you're reporting on the results of a computer simulation, screenshots are really helpful. So depending on the computer you're using, you can use, if it's a PC, you have the snipping tool. If it's a Mac, you have your own way of capturing screenshots. But I strongly encourage you when you're describing your results of a simulation to include appropriate screenshots. Something that came up in one of the homework problems last week and will come up again and again is that when you are doing simulations, you need to adjust the time scale over which you're doing the simulation so that you have something that's appropriate. In the bottom, we have an SIR model of an epidemic. We're changing one of the parameters. It's going to change the time scale of the epidemic. In this case, I've kept the time scale for the x-axis the same, 0 to 20 in each case. What we'll see is that in the middle case, everything looks about right. I can see the appearance of the infected population and then the gradual disappearance of the infected population in favor of the resistant population. If I go to a situation where the epidemic happens more slowly, I don't see much of anything happening. It's just starting at time 20. And if I go to a situation where the epidemic happens faster, where the rate of infection is faster, I have a sharp peak around day 1, and I can't see that either because it's all compressed. And so I have to look at the outputs of my simulation and ask the question, are my scales on my x-axis appropriate? In the case of the one on the left, 0 to 20 isn't showing me the information I need because the epidemic is over by time 10. In the case on the right, the epidemic is just getting started at time 20. And so in the case on the left, I would adjust my x-axis to be 0 to 10. In the case on the right, I'd probably run from 0 to 50 or 0 to 100. And so as you run simulations, you have to ask the question, are the time scales and life scales you're running over appropriate, and adjust accordingly. When you use longer time scales, you may have to increase the number of points that you use in the simulation. As you remember, we have to simulate initial time, comma, final time, comma, number of points. If you make the final time very long, you might have to add points to the simulation to keep the numerics in reasonable agreement with reality. But in general, I think people managed pretty well jumping into these problems early on. If you didn't submit a homework, please do. They're important. A few people didn't fill out the intake questionnaire for the course. I remind you that you get points for filling out the intake questionnaire. So there's no reason not to get the extra points for doing that. So please do. Okay, so I'd like to move on now to student presentation of the papers. One of your assignments for this week was to find 5 or 10 papers that were potentially interesting to you, and then prepare briefly a short presentation, a slide or two, to explain the 2 or 3 most interesting papers that you found. Does anybody want to get us started here? We'll go around the room and everybody will present. Otherwise, I'll just call on the person at the top right-hand corner, I guess. Benjamin?
Yeah. So far, I have only like really looked through the paper that I submitted for last week.
Okay, do you want to?
So do you want me to go over that one?
That would be fine. If you want to screen share and tell us about that, that's fine.
Do you want me to navigate to that paper?
Yes.
For the screen share? Okay.
However you'd like to present it, it's fine. If you need more time to get set up, I can go to someone else and come back.
Yeah, I might need a couple minutes.
Okay. I think it's important that everyone listen to each other, because the point of these presentations is to see whether there's somebody else who's doing something you're interested in, to team up. So Drew, do you want to take over?
Yeah.
Okay.
Okay. See, I've got 3 different papers I was looking at. And so this first one is about they're modeling, it's kind of an old paper, which, I mean, I find it kind of interesting. I'd like to look at it, but it's modeling these 2 proteins that are involved in the cell cycle that they form a promoting factor for the cell cycle, and it results in like different states that the cells could go into, like growth in like embryos or normal cells, or like halting the cell cycle. And I don't know. There was a lot of math behind the paper. It was kind of above me. It's going to take me a while to really process how they build the model, because there's a lot of I don't know, a lot of inputs that they put into it. But the other ones that I was looking at, this one was for COVID-19 exit strategies with lockdowns. I thought this one was cool because they're looking at different lockdown strategies, and I think the most useful image to describe what it was is this, where you can see they're trying different they're modeling different ways to have lockdowns and work for people, and they use, you know, statistics from population density and other stuff to predict if there's going to be outbreaks and an overall decline in the virus. And a similar paper, this is the one that I had on my homework one, is I didn't have anything for this slide, but this one was modeling it on a cruise ship, just seeing what lockdown procedures are necessary to prevent an outbreak in a small controlled environment with a high population density. And the thing that they found was that it basically needed really, really strong restrictions with their model. But yeah, those are the three that I'm really focused on right now.
Okay, thank you very much. Let's see what other people are coming up with and see if there's some shared interest. The cell cycle model of Tyson is one that we've taught in the class in the past, when the course was more focused on chemical reaction networks. That's a very beautiful model, and there are a lot of extensions. That core model that he developed in the late 80s, early 90s was then elaborated to try to understand the details of cell cycle. And so if you're interested in how cells actually regulate cell division, that's a would be a great piece of sort of classic mathematical biology to play with. The infection models that you talk about are quite interesting. I remember when that paper of Uri Alon's came out, the one on the lockdown exit strategies, and it's a case where perhaps events have overtaken the model. That was released, I think, in April or May of 2020. It seems an age ago, but that's great. Okay, good. Well, let's move on. Who would like to go next?
I can go next.
Okay, thank you.
Can you all see my screen? Yeah, so the theme that I wanted to explore was a deep learning surrogate for biological models. The reason we would like to use deep learning here is like there are three main reasons. First is the fact that any learning that happens using deep learning models is unsupervised. So there is not going to be any prior knowledge bias that basically around the biological model that we are trying to develop. So we could probably run into some very useful and very counterintuitive like conclusions if you do that. And second is that it's obviously where all the technology is headed these days, and it makes things work way more faster. Okay. Yeah, I was just checking if I was on mute. I'm sorry. Yeah, so it helps it helps speed up the whole process of modeling. So the papers that I kind of worked on are what's called indoor air and stock prediction with ML. So indoor air is basically a paper that explores viral kinetics of the spread of influenza infection in an indoor setting. And it basically lays out a bunch of models that help us track it. And the thing that I was interested in here was basically the OD equations that basically lay out the spread of this infection. I'll be getting to that later. And the second paper is basically stock prediction with machine learning, which basically discusses in very brief walk forward modeling kind of neural network. So yeah, so in the first paper, you can see towards the right that there are like 4 ordinary differential equations given. And they basically define how the rate of change of infected cells, cells that are infected and are producing virus, cells that and so on. So basically, they're like producing virus infected cells and virus titers. Virus titers basically means that it's the lowest amount of like lowest amount of the nasal discharge which can infect a person. That's what virus titers mean. So these basically what we're trying to do here is we're trying to come up with a training data based on the ODE equations. We are trying to, and then we are trying to split that data into a tested and a training set. Using the test set, we are trying to train the neural network and help it make predictions. So the second paper basically gives us an idea about how we could go about that. So as you can see here, M1 through M5 basically is the prediction that we are like is basically the endpoint of the training data, and the part that is highlighted in blue is basically what we're trying to predict here. So the first homework that we did in the last class, we know that we get like a time series data frame with like with the values of all those 4 variables at every given point of time. So the input to this model would be those values like starting from 0 to any given set of time, and we would be probably predicting t plus delta, like what would be the value of those variables at t plus delta. So yeah, there's some minor data preparation work involved that we would be taking care using pandas, and we would be making these predictions. Yeah, so the underlying idea here is basically that if we could come up with a surrogate model that can do biological modeling, then it basically opens a lot of doors for us, and like we could extend it to biological models that go way beyond the ones that can be predicted using ODE. We could probably look at a whole host of problems that we can solve at that point. So yeah, thank you.
Okay, that's great. I mean, in principle, you could pair with anybody, and whatever their model is, you could try to do a surrogate for it.
Yeah.
So that would be nice. That's a nice complementary set of approaches. That's a good example to start with. Okay, who would like to go next?
I don't think I have as in-depth a presentation for what I've got so far, but I found the thing.
That's okay. I mean, the reason we're going to have sort of weekly assignments on this and weekly or every other week check-ins is because it takes time. And so don't worry about not having everything finished. Go ahead.
Yeah, okay. So the paper I went through was modeling the effects of different treatments based on their theoretical effectiveness and the time at which they were administered, which I thought was interesting because we've been at the point that we are with COVID, we are very deep into into prevention as treatment, and this was modeling for cures before the cures had been put through clinical trials, right? Which was something that I had never even thought about doing, but it makes a lot of sense as a saver for resources. And what they found was that in order for a COVID-19 treatment to be effective in ending the pandemic, it would need to be applied very early on, and it would need to have high effectiveness, which is something very difficult to do, something very difficult to develop. But on the bright side of it, they also found that if those prerequisites are met, that the virus's known ability to adapt would not be a concern.
Sorry, whose paper is this? Can you open the author list? Okay, this is the Perlson one. Okay. So yes, that's because there were two papers. There was one paper that did a little bit more detail on the molecular basis for the hypotheses. So there was a paper by David Oda from Minnesota that was trying to look at drug repurposing. And then there's this paper, which is a nice one. Perlson is one of the great experts in this area. And it's a nice paper. As you mentioned, the problem with all of these antivirals is that by the time you have symptoms, it's usually too late to use the antiviral, and so that's a problem that we face with any antiviral strategy. But yes, that would be. So this is I'm trying to remember. This has been a while. This is the two compartment model, as I recall. They have a model of the upper respiratory system, the lower respiratory system, if I remember right. For this.
Yes, I believe so. And they were the models were of the amount of viral load in, I believe.
Okay, so that's certainly one. That's certainly one that we could pursue. TJ, you know that paper pretty well.
I do. I'm quite familiar with that one. It's a nice choice.
Yeah. We've had some conversations with Alan about it and some of the other people on the author list. So if somebody decided that was a paper to work on, that would be interesting. And it's certainly one that could be, you could do the AI surrogates for. It could be interesting. Okay, great. Okay, so who do we have left? Ali. Colin, do you want to go next?
Yeah, I could go. I don't have a presentation, but I at least have the main article pulled up that I wanted to look at. I looked at a few though, and I'll explain a bit about them. Let me share my screen. Okay, this is the second article that I had looked at. The first one was an evolvability model actually by Draghi and GP Wagner that I looked at briefly. And that one essentially looked at random mutations or evolvability rate within certain populations in a simulated environment. And it wanted to basically see if the survival rate of that general population would increase or decrease based on higher evolvability, which is just this simulated model for the real world scenario where genetic mistakes occur when DNA formation is happening in certain individuals, and that'll lead to certain genetic changes that could basically also be used as a natural selection model. And they did find that for the most part, this increased evolvability rate would lead to more population success in the future. I can pull up I can at least send out the article, link to it in a bit. But this one that I looked at more so, I also looked at for my first assignment, and it was another viral network model, but it had more to do so with the control strategies used to prevent COVID-19 and to at least decrease the spread of COVID-19 throughout certain communities. And what this did is it looked at the containment methods that were possibly used in most cities, at least, and most like real ideas. And it looked at a place specifically in Haslemere, United Kingdom, and it used the exact statistics and data from the people over there. I think there were 468 individuals with a certain number of social links, which is basically just to say there was a certain number of interactions per day for every individual there. And it basically looked to predict percentage of total population infected after a certain number of days, given each different method that was used. And the methods that were used were where there was no containment, there was no source of control for COVID-19 infection. There was also isolation of symptomatic individuals only, meaning that they were quarantined and basically cut off from the rest of the population. They also use primary contact tracing and secondary contact tracing as their third and fourth models. Secondary is just where you trace the contacts of the contacts. And what they found eventually was that obviously with higher rates of symptom tracking and contact tracing, they were able to mitigate the actual number of total infected individuals, because you could be either infected, not infected, or recovered. And in their model, if you were recovered, you couldn't be infected again, which is not perfectly accurate of COVID, but I think it's still a pretty good representation of a general control strategy for the virus that's obviously still very prevalent today. And that's about it with that. And that was the second one I looked at, but I didn't have a third right now. I was still kind of searching around for others.
Okay, that's great. I mean, that's a fascinating piece of work, and it's a beautiful model. It's not something that the particular model structure isn't one that lends itself to using tellurium. And so it's not that one couldn't work with that, but this is in a sense more of a dynamics of networks model rather than a dynamics on networks model.
Oh, I see.
Because you see that you have your individuals who are connecting, and those connections are stochastic. I mean, again, it's not. I'm not saying you couldn't do it, and I'm not saying it's not a fascinating problem, but the kinds of modeling we're going to focus on in the class part of the class, in the lecture part of the class, are more directed to situations where the network architecture is fixed, and you then have dynamics on that network architecture. This kind of is fundamentally an agent-based modeling approach, and it's very interesting, these kinds of Boolean models. But we should probably think about what we do with it if you were going to try to work with it, and see what would be practical. Nothing wrong with trying to replicate this paper. It just they'll wind up doing everything in Python and not using the antimony and tellurium very much. It doesn't mean it's not a worthwhile thing to do. and the evolution model I don't know about. So that might also be interesting to look at. But that's fine. Anything more you want to talk about now, or just show us that for the moment?
No, that was just all I wanted to show for now.
Okay, great. Okay, I think we have two people left. As Ali.
Hi, can you hear me okay? Okay, great. I also don't have a presentation, and I'm a little bit behind just because I had like some issues, like my laptop broke and stuff like that. But I do have like one article I'd like to share. I have it pulled up. Let me find it. But the title of the article is it kind of details of how neutrophilic cellular traps the regulation of lung cancer invasion and metastasis. So basically, the beginning of the article kind of discusses how the traditional role of neutrophils is to, provide an immune response. And like one of these immune responses could be to like defend against cancer and tumors. But one of the problems that we face, I think, is that the neutrophils are actually hijacked by the cancer cells, and then the neutrophils then actually allow the cancer to spread even more. And so there's like the article kind of details a mathematical model that takes into account like the densities of the tumor and the neutrophils, and the concentrations of signaling molecules and structure, including neutrophil extracellular traps or nets. And if you like, I still kind of need to like go through it more in depth, but it has some diagrams, and then it kind of goes through what the actual mathematics of that would be in vitro experiments. There's like some more diagrams, and there's like all of these sorts of factors, and there's actually like a huge table further down, if it'll load, kind of detailing all of these factors and how I think they can affect like how a tumor will spread. And then the main overarching purpose of this computational model, I think, is to study how the tumor spreads in order to create better anti-tumor medications, if you will. I don't know if medications is the right word, but yeah, that's mine.
Okay. So that's an important problem and an interesting model. I didn't know that particular one. Again, a little bit like the last paper, the model that you're showing there is a full three-dimensional agent-based model of cancer invasion. And so if it were the spring semester of this class, where we do spatial agent-based modeling, that would be a perfect topic. For this semester, the question would be if we can find something that focuses on population dynamics without the spatial component. And so if you're interested in understanding cancer progression, for example, or cancer suppression, metastasis, there are many papers that address that in a non-spatial context, in a population-based context. And I think probably starting with those might be a good idea before you jump into these very complex spatialized models. And I realize that sometimes it's frustrating in the sense that if we focus on these simpler models in this class, you may not get to the level of predictivity that you're interested in. But there's some reasons and advantages perhaps to doing that. But But as a topic, I think this question of lung cancer invasion metastasis is a great one. And we can definitely pursue that. Am I connected?
Yes.
Because I'm getting some very peculiar Zoom effects. Let's keep going for the moment. I may wind up having to leave the meeting and come back in because Zoom is gradually deleting. I can't mute, for example, and the buttons for the control of the meeting have disappeared from my screen, among other things. Plus, most of the participants are blanked. So I'm not quite sure what's happening with Zoom. My guess is Zoom did some kind of update to help us all out at the start of the semester, and we'll have to live with that. So, all right, Juliana, are you still there?
Yep, I'm here.
Okay, because you've disappeared from the participant list.
Huh.
There's something rather odd going on with Zoom. Okay, and I guess the last presentation is it Delaney? Are you there?
She said she's having some Wi-Fi issues in the chat.
Okay. Well, I can't see. I can't get the chat to work. So I'm going to exit. See if I can make. That brought that back. Giuliano? Huh.
Yep.
You're a co-host, right?
No, I don't see it by my name. Same for TJ.
Well, it goes figure that when we decide that things would work better when we did it this way, it decides not to work. And let me see if I can get. I don't want to end the session. So let me see if I can. No. Well, this is a new problem. My Zoom session has died. Sorry, everyone. If you get thrown out when I try to restart the Zoom, please log back in. It's not a problem I've ever had. Well, that seems to fix things for me. Can people still, are people all right for their Zoom?
Yes.
Okay, sorry about that. I don't know what happened. So, and she's, Delaney, are you able to present now, or do you want to do that later?
Could I possibly present next class? I am having a lot of technical difficulties today.
Well, I guess I am too. So it's, I can't say anything. Okay. What I would
Thank you.
ask people to do is I think it's important, look at each other's presentations and think about whether there's somebody you'd like to work with. And also, please make an appointment to meet with me and Giuliano and Josh together to look over the papers one-on-one to see how practical they are to be a project topic. If the topic is not, if the particular paper won't work because there's something technically difficult about it, we can then do some Googling together and see whether we can find a paper that's in the same thematic area and same content, which is computationally a little bit easier to work with. So I hope we're able to find something that gives you the domain interest that you want, but it's also something that's achievable. I rarely will tell people you can't do something very complicated, but based on many years of experience, I would recommend picking things that are relatively simple because the semester is not so long. And so when people are very ambitious in what they try to do, sometimes the result is frustration because people get stuck. On the other hand, if you want to be very ambitious, I'm not going to tell you not to. Okay, thank you everybody for the presentations. We'll revisit these things next week. If you heard somebody else presenting something you're interested in, please hook up with them and talk together. And do contact me or set up a meeting before next week so we can talk. Okay, so I'd like to come back to where we were last time. We had some basic computational structures, some very simple ones, using some Python functionalities that we're going to use again and again. So one of the things that we were doing was, for example, our simulate function will return an array consisting of a column of the times, followed by columns representing the variable variables. And we wrote a simple function that looked in one of the columns and asked the question, when had the number of uninfected individuals decreased by half from its initial value? And then it recorded the time at which that happened and the value. So that was this first loop using a while loop as a iterator. We also thought about how to do parameter scans. In this case, we used a parameter scan in the exponent of the population. We reset the initial population in our model, remembering that we actually had to do that twice because of the way antimony is parsed. Ran the simulations, and then plotted them on top of each other using this show equals false command to have the graphs overlay each other, and then the te.show to actually display the result. A little bit more sophisticated way to achieve something similar was a parameter scan using the NumPy vstack option. In this case, we had to create an empty array to hold our results. And then we ran the same simulation and accumulated, in this case, the result value and the mid-time value. And in this case, we didn't plot it, we printed it So these are three loop structures, three iterator structures, one using a while loop, one using a for loop, one using both a for and a while loop, and different ways of manipulating the data that we get out of the simulation. Last time we stuck with the very simplest possible model of an epidemic, that we have people who are uninfected, people who are infected, the infected people infect the uninfected people. We don't know anything more about them, so we assumed that the rate of infection was proportional to the probability of encountering an infected individual. And that gives us a simple exponential form for our results. And the last thing that we did at the end of class was to write a parameter scan that sweeps gamma, that is the infectivity per encounter, and then ask the question, how does gamma change in gamma and the infectivity of the virus change the time at which half the population becomes infected? And that was the last exercise that we had last time. And I think we should probably start now with that. And so if people can get Tellurium running, you may have this simulation already running from last time. If not, we can do it together. And so let me get my screen set up for that, and we'll go from there. So people, please try this exercise on your end. If you've done a more sophisticated model like the SIR model in the homework, then in a sense, this is a downgrade of that model, but you can definitely use that as a place to start. So give me a second to get things running here. Okay, so what we had in the last time, do people have this, or people have been able to find what we did last time? You should have the downloads from last time. We did this in class. If not, you can find it in the student materials folder or in Canvas. So if anybody's having any trouble finding it, please speak up and we'll help people out. So I'll give people a minute to find things from last time. And if anybody needs help, please speak up. We'll do our best to help people out. So this is not the answer to this problem because I haven't changed the value of gamma yet. This is just the base model that calculates that mid-time, which is what I needed to know how long it takes for the simulation to reach half of the population infected. And I can do this now in parallel with you. I'm not trying to rush anybody. And I'm not guaranteeing that I'm going to do it better than you do it. But let me just try. We've loaded our model. We'll sweep gamma. Gamma is, in this case, let's see, what was our value of gamma? 0.1. Can't be bigger than 1. So our value of gamma has to be a number between 0 and 1. So I'll do for gamma 1. Gamma is a reserved word, so you have to be careful about it. In n-pi.a range, and let's say 0.1 to 1, steps of 0.1. We'll indent everything here, I hope. And before we do our simulate, we have to reset. We have to reset our initial conditions. And we have to reassign the value of gamma. I said S, it should be N. I think that should do it. Save my. So I forgot to overlay the plots. So let me do the plot overlay. Okay. So let's see if people are able to get something that looks a little bit like that. I didn't do a very pretty job of it. What you see here is that I've looped over the value of the gamma parameter. I've reset my simulation because each time I run it, I have to start over. I've overloaded the value of gamma in the simulation with the value I want. I run the simulation. I plot it with show equals false turned on so that it doesn't, the plots overlay. And then I calculate the time to get to half population, print it, and then eventually I show the results. Since S and I are just 1 minus each other, probably I don't want to display both S and I. So I'll use the option that I have to select only a particular column of my output. So I'll select the time column, and the number of uninfected. I'll get something that looks like that. So let me try again, see if people are able to get that. There are other ways you could achieve the same thing, so don't worry if you had a different approach to solving. Yes, that's right. This was called SI Model Lecture 2 exercise. Stack midpoint A. It's exercise 1, stack midpoint A. I'm sorry, I didn't see the chat. The chat window was hidden, so I didn't see it immediately. And if anybody needs help getting this to work, that's okay. If you've gotten through it already and you're looking for something else to do, you might think about how you would plot gamma 1 versus midtime for your simulation. If I look in my printouts here, I see that it takes approximately 13 days to get to half infected when the infectivity per encounter is 0.1. And that goes down to 1.4 days when the infectivity per encounter is 0.9. I'll let people keep working on that. I wanted to come back to something that I rushed at the end of class last time, which was how to do some graph annotation. The clear result here is that the more encounters that each person has, or the more probability of infection per encounter, the faster the epidemic spreads. So social distancing and mask wearing both have the same effect. In one case, we're reducing gamma, the infectivity per encounter. The other case, we're reducing N, the number of encounters per day. And of course, as we mentioned before, in a simple model like this, everybody gets sick. Eventually, everybody gets infected, no matter what you do. It's possible, actually, to solve these equations analytically. They're fundamentally just exponentials, nothing very fancy going on. The number of infected individuals is essentially the exponential of n gamma times t. There's a normalization constant. At short and long times, the number of infected individuals grows exponentially, simple exponential. You don't have that saturation. And the time scale for the growth of the infection is 1 over n gamma. So we should be able to see that if we plot gamma versus the time. Something that we might want to do is adding some labels to our graphs. That's not very exciting coding, but it's useful to be able to add labels. The plot functions that we're using here, there are a number of possible plot options that we can use. In this one here, we used m.plot, which is the built-in, one of the built-in methods for plotting. There's also the TE plot array option. Somewhat confusingly, the options within plot and plot array don't have exactly the same specifications. And you probably need to look at that Python, the Tellurium cheat sheet that we handed out, or go to the Tellurium manuals if you get confused about what the options are. We can color our plot by turning on reset color cycle equals false. That was something that we were just doing at the very end of the simulation last time. And now that I think about it, I keep typing it on. Now that I think about it, this only works here for the, we have to use the plot array function. It doesn't work for the default plot. When we switch from the default plot to plot array, we have to put the name of the array we're plotting inside of our plot command. And let's see what I did wrong. Typo, probably. Which one did it crash on? Title. Okay. As I say, unfortunately, the axis labels and things are different in the two function calls. Let's see if that works. No, doesn't want to work. Let's see. The plot array S title population. Get rid of the title maybe. It's not going to be my day, I guess. Giuliano, do you see what's wrong?
Trying to spot it.
Yours worked? Let me restart my kernel and see if that's. Okay, so I don't know what it was plotting it. So why was it not? The issue is the display here. Are other people having it work? Let's see, run. Why is it not? Very interesting. If I take out the show equals false and run it, it displays all of them. There's the one with the show equals false and the color cycle turned on. And then it displays them individually. So the problem is the t.show is not. So why don't we, instead of doing it that way, we'll do I'll plot the last one again by hand. There. All right. I'm not sure why that behaved in this anyway. Okay. So what I was trying to show you last time at the very end of class was how to get colored bars, lines, which was probably not worth the effort just now, using this reset color cycle equals false. And now what I'd like you to do is add labeling so that we know what the value of the gamma parameter is for each one of the colors. And the basic way you do that is you have an option in the plot called labels. Labels can either be added one at a time when you iterate through the sequence of plots, or they can be stacked in a list and plotted all at the same time. The argument of labels is a string. And so to make that to make gamma into a string, you have to use the Python function str, which converts a number to a alphanumeric string. So why don't you try doing that? Let's add the label here. So we add labels equals, and then we're going to add the values. So we'll say gamma equals. So we remember what our variable was. And then we'll add to that, because it's a string, str of the value that we just set, which was gamma 1. Let's see if that works. So we get something that looks like that. Are people able to get that? Let's see. Let me make this a little bigger. If people have caught up with that, one of the things you'll notice when you do string conversions is that occasionally you'll have a lot of trailing zeros, which isn't wrong, but isn't very pretty. And there's a Python function called round that truncates to a given number of decimal places. So if you do round, the name of the variable, and then comma, the number of decimal places, you can clean this up. So instead of saying string of gamma, you'd say string of round of gamma, comma, 2. And then I have a wrong number of parentheses. Now what happens? There. And that'll truncate it. So try that out. Again, I don't expect people to memorize all of these functions. but it's useful to remember to learn how to look them up. So if you type into Google, how do I round a number in Python to x decimal digits, it will pull that up immediately. So question, what's the difference between the TE plot array and the mplot call? When would we use which? That's a good question. M.plot was designed to be the simplest possible thing for plotting. It tries to guess the things that you want to do. So it labels the x-axis, the y-axis, it puts up the key automatically and so on. The only disadvantage of m.plot is that it'll only work on the output of m.simulate. It won't work if you've built an array to do, say, the x-axis being the parameter value and the y-axis being the variable. And so you have to use the plot array or one of the direct matplotlib functions if you want to plot arrays that you've built yourself. The other disadvantage of the m.plot function is that it's more limited in terms of the settings that you're allowed. And so in general, I use the plot array most of the time. Any other questions about this? I know between the Zoom problem and then some of these other things, it's not things never go smoothly. I guess it's life. Okay, so here's that result with the rounding put in, and so now we have a display that we can use. Again, if you look in the manuals for Tellurium, you'll find details of all of the different settings, how to do logarithmic axes, how to put tick marks in, and as you want things to get more and more complicated, you wind up getting more and more to matplotlib. Matplotlib is very powerful. It's not the friendliest package in the world, and in your projects in particular, you may find that matplotlib is a bit of a bear, because often in these projects for the applications, you'll want to have your graphs laid out in particular configurations. Matplotlib allows you to do very complex layouts, but the syntax for some of those isn't so obvious. Okay. There's another way of doing labeling, and let's do that quickly as well. We're going to be doing the same basic idea here, but instead of using the plot overlay that we've done here with the plot false, let's do the same thing using an H stack. So we're actually going to build our array line by line. So to do an H stack, what we're going to do is we're going to create first an array that's going to hold the, well, I have slightly different example here, but I can do it anyway if you want. I'm trying to think about the best way to make this work. Okay, so let's do this with an H stack. So if you remember H stack, what we're going to do is we're going to create a column vector that contains our times. And then we're going to put on the right-hand side of it each time a vector which contains a column vector which contains the values that we want to plot. So for an H stack, what we would do is the following. We would start out by creating some placeholder array that we're going to use. And we'll say that that's m.simulate. There are more elegant ways to do this, but it'll work fine. 0, 20, 100. And then we're just going to put time in it. And so if I plotted that, all it is the x-axis is the time positions. Now we're going to do our same loop for gamma in A range, reset M gamma. Now when we do our simulate, we're only going to give the variable. So we only want the column with the variable. We're going to get rid of our TE plot now. We'll do our simulate. And now we're going to do a H stack, which is N1 equals numpy dot H stack of N1 comma S. And you have to put those in. It's actually a list as an argument, so you actually have to put that in. And so what this is going to do, each time we run this, it is going to append to the right the column vector of the variable. We're going to do the calculation of the midtimes anyway, and we'll plot this at the end. So let's try that. Except now we've accumulated everything in N1. Oops. X index is out of bounds. Oh, because I didn't, this won't work here. Sorry. Can't do that calculation for the moment. Just do it. Just do it. Okay. In this case, this was really answering this question about plot array versus mplot. In this case, we've created N1, which has all of our columns in it. So if I print, let me print N1 so you can see what it looks like. N1 has the first column is the time. The second column is the value of all of our variables, our variable S versus time for the first value of gamma, the second, the third, the fourth. In this case, if we want to label this, we have to build a stack of labels. And the way we can do that is by creating a placeholder for our labels. So we'll create a placeholder list label equals blank. And then here, when we add our variable column array to the right-hand side of our array, we're also going to append to that list the thing that we would have plotted in our other method. So we'll do label.append, add to a list. And now we have to do exactly what we typed before. We want our gamma equals, and it's still a string as an argument, str of round of gamma 1, comma 2, and that should do it. Now we want to plot, when we have to add to our plot labels equals label, and we should get our result here. So we've recovered the same thing. So we had two ways of doing the same thing. And depending on what you're trying to do and how elaborate you want your plotting to be, you'll find one or the other of those may be more convenient. Again, this is for the moment, just a little bit of a first experience with this. As you go through the exercises and homeworks and your project, I'm sure you'll come up with ways of doing plotting that you find more appealing. Okay, so the next thing I'd like to talk about is something that we've mentioned a couple of times, but haven't done, which is that so far we've always plotted variables like S or I, but when we use the simulate function with a named variable like this, we can also tell it to plot E1. And what E1 will do is it will return the value of this argument here, n times gamma 1 times I times S over S naught. That is, it will return the rate of change at which S is converting to I. And so that's a useful thing to be able to do, because you might want to know what the rates of things are as well as the values. So effectively, you get two things at the same time. You get the value and the derivative in time for free. So why don't people modify this plot here so that you plot the rate of change of infection, the number of new infections per day, rather than the number S. I'd like everybody to try to make that change. Is it clear what the exercise is? If people had trouble catching up with what I did here in terms of changing the plot, you can use the plot that we had before. You can use the old version of the plot. You don't have to switch to this H-stack formatted plot. You could use the other one. Remember that when you run simulate in the way we've done it here, you're putting out a single column, and the argument, the variable that's being plotted is whatever you name in that column. Does anybody have it? Somebody says they've got it done. Would you like to show what you got? I can't tell who the person was who said in the poll that they were done. So whoever is done, why don't you show us what you did? I can stop sharing and let you screen share if you like. Was it Drew? Was it Drew? Were you the one who said that you were finished? Nobody wants to volunteer? Okay.
Drew says that his mic is not working and that he can't share screen because it's on another laptop.
Ah, okay. Sorry about that. Okay. So to do this, some of this may feel awkward, but once you've done it a few times, I hope it feels less awkward. if I want to plot the time derivative instead of the value of S, all I need to do is change this variable from S to E1. And now it will plot the time derivative, the value of E1, which is the rate of change of S as a function of time. And you see I have lots of peaks here. And you'll also see that they get a bit jagged. If I zoom in here, they're a bit jagged at the top. When you see that kind of jagged interface at the top, that means that the number of intermediate points you're using in your simulation isn't enough. And so I could make the number of points here larger. And let's see. How do I do that? I have to change it. And now I'll get a smoother curve. So one of the things I'd like you to practice a bit is manipulating arrays, assembling them together using VStack or HStack, selecting columns that you're going to plot out, and beginning to familiarize yourself with these kinds of simple operations. Okay. So here we have the rate of new infections versus time. And I'm sure over the past year and a half, you've heard statements in the news with some frequency about the rate of new infections per day was such and such. And so that's the specific reason that I wanted to plot the rate of new infections per day. If we look at these curves, we already see something a little bit interesting, which is that as we change gamma, remember gamma is the probability of being infected when you encounter an infected person. If gamma is small, the rate of new infections peaks late, and the rate of new infections is also spread out over a long period. Even though ultimately everybody is infected in each one of these simulations. So changing gamma doesn't change the total number of people infected because everyone is. But what it does change is it changes the maximum rate at which we're infecting new individuals and the time it takes to get everybody infected. As gamma gets bigger, as the probability of getting infected when you encounter an infected individual goes up, the time to get everybody infected gets shorter, and the maximum number of people being infected per day goes up. And so this is precisely what is meant when they talk about flattening the curve. In this case, the curve is always this Gaussian shape. However, as we make gamma smaller, the probability of infection per encounter smaller, the curve gets delayed to later. It gets spread out, so it takes longer to go through, and the maximum value goes down. Now, why would that be of use? If everybody's going to get infected anyway, you might say, why do you care about the shape of the curve? Well, if the number of people hospitalized is proportional to the number of new infections, which it more or less is, and your hospitals can only handle a certain number of people per day, then reducing this curve means that you can treat more people. If everybody's getting infected at once, you overload the health system. If people get infected over a longer period of time, your health system has more time to accommodate. At least that's the theory of flattening the curve. And so this is actually a simulation that shows flattening the curve in a very literal way. You're taking this curve of new infections and you're spreading it out horizontally and you're reducing its amplitude vertically. Okay. Were people able to catch up with that? If not, we can take some more time and let people catch up. Okay, so one thing we can do if we're at home, which is harder to do in person because of the university rules at the moment, is we can take a break. So you don't have to sit continuously for 3 hours. So why don't we take a 5-minute break? People who are still working on the problem can continue to work on it. I'd like to continue now to think about what results mean. And I'd like to remind you of something we talked about last time, which is if I'm asking you to think about or describe what you see, my suggestions are always the same. When you're looking at a time series, start out by looking at what happens at short times. Ask, what's the value at short times? In this case, the value of the rate of new infections is nearly 0 at short times. The number of people who are uninfected is nearly 1 at short times. And the number of people who are infected is very small. And then you ask the question, is the slope in time positive or negative or 0? Well, it looks like it's almost flat, but that we know that there are at least some new infections, although very few. And so the slope is increasing. The rate of new infections is increasing, but very slowly at early times. Then we look at the opposite end. What happens at late times? Well, at the end of this situation, everybody has been infected. There are no uninfected individuals, and so everything stops. So at very long times, the rate of new infections is 0. The rate of uninfected individuals is 0. The rate of the number of infected individuals is the number of individuals in the population. And then we want to look in the middle and ask the question, when are changes most important? In this case, that would correspond to the slope, the rate of new infections being maximal. And here we see very clearly the rate of new infections is maximal in this plot around 13.7. Another thing that we might want to think about is whether the rate of increase of the infections and the rate of decrease of infections are the same. In this case, if I draw a line down that 13.75, what I see is that this curve is effectively Gaussian. It's symmetrical. The rate of increase of infections and the rate of decrease of infections are the same. We'll find when we go to SIR models that that's not true anymore, that they're not symmetrical. And so if I asked you, tell me what you see, I'd ask you to walk through these 4 steps and describe how those steps play out in the time series that you see. So now we can look at this and ask the question, does this SI model describe what we see for COVID? And now we'll go back to the early phases of the epidemic. And if we go to something like Worldometer and look at the rate of new infections in Italy and the UK, what we see is that the leading edge of this isn't too bad. We see the number is very small. It grows roughly exponentially for a while, that it saturates. It seems to saturate a little bit faster than we would get for the SI model. The peak seems to be a little bit broader, but then we also see something that isn't the case in the SI model. In the SI model, the rate of increase of infection, the rate of decrease of infection are the same. In the case of Italy and the UK, the turn on of the infection, the incidence of the infection was pretty fast, but we see that the clearance of new infections, the rate at which the rate of new infections goes down, is much slower. If we look just as a ratio, the time for the infections to go down again was about 4 times the amount of time it took for the infections to turn on in the first place. For the UK, the rate of decrease of infections was even slower than in Italy. That's probably because social distancing was more effective. The lockdown was more effective in Italy than it was in the UK. But we can't get an asymmetric graph with an SI model, and that means that we cannot match the population incidence of the disease using an SI model. So now we're going to go to the next level of complexity, which is not much more complex and which was already in the homework, which was the SIR model. And so the basic idea of an SIR model is that this is an infection that where your immune system clears the disease. An SI model, once you have it, you've got it. So that would be like HIV. In an SIR model, your assumption is that once you've had the disease, you get rid of it. And it's going to assume to begin with that there's no reinfection. Unfortunately for COVID, we know that's not true. I've probably mentioned this already, but back in December, I already knew 2 people who'd had COVID twice. So it looks like if you're lucky, having COVID confers about 9 months of immunity, maybe 6 months is more typical. And so we don't really expect that our recovered individuals never get the disease again. And we could certainly add to our model that reinfection probability. But to begin with, we'll start by assuming that once you've had it, you're immune. Smallpox, mumps, and rubella are examples of diseases which you don't usually get twice. This was in that textbook that I mentioned, the Brower textbooks, chapter 2.4. The model is traced back to by Brower back to 1927, long before computers, by Kermack and McKendrick for modeling epidemics. So in this case, we're going to assume that individuals now are in 3 categories: susceptible, infected, and recovered. The recovereds could also be dead, because if you're dead, you're not going to get infected again. And so you might want to split those populations into recovereds and deads, but for the moment, we're not going to do that. So we're going to say that susceptible individuals become infected when they're exposed to the disease. That's not going to look any different from our existing SIR model. And then once you're infected, you could recover. In principle, the rate of recovery could depend on the number of susceptibles in the population or on the number of recovereds. In practice, your medical recovery is going to depend on your own immune system. And so in general, it's going to be a function only of the number of infecteds. So we'll keep our original model for the infection rate. Probability of being infected is the number of encounters you have per day, the probability of being infected when you encounter an infected individual, times the fraction of the population, I over S naught, that's infected, and then the total infection rate, that's per person. The total infection rate is just S times that. And now we're going to assume that people recover on their own. It's what's called first order decay kinetics. We'll say that they recover at a rate mu, which means that they stay sick typically for 1 over mu days. So if mu were 0.1, that would mean they stay sick for approximately 10 days. Now it means that since each recovery is independent of the others, the recovery rate is just mu times I. So now we can write our population model. S goes to I, the rate n gamma I over S naught times S. I goes to R at a rate mu times I. And that's the SIR model that we worked on in the homework. Let's define a parameter beta, which is the total infectivity of the population. Each infected person infects n gamma S over S naught people per day. Notice that this is a function of the number of uninfected individuals, so it will change. It's not a constant. And they stay infected for approximately 1 over mu days. If I'm an infected individual and I infect n gamma S over S naught people per day, and I'm sick for 1 over mu days, then the total number of people I'm going to infect will be n gamma S over S naught over mu. I can extract from that, because that's a function of how many people are uninfected, the typical number of people that one infected person infects, and that's n gamma over mu, and that's called R naught. And R naught has become a term that people see in the news all the time. We've got to reduce R naught below 1 to get rid of the infection, get rid of the epidemic. And this is the number that everybody talks about. N is the number of encounters per individual. That will change depending on social distancing and personal behavior. Gamma is the probability that an infected individual infects another person when they encounter. That would be changed by something like wearing a mask. On the other hand, it's also a property of the virus itself, partly, because some viruses spread easily and some don't. So for example, the lifetime of the virus in the air or on the surface will change that gamma. Mu is the amount of time over which you're infectious, which is not exactly the same as the amount of time you're sick. You could be infectious for a long time and not sick for a long time, but more likely, actually, you're more infectious when you're not feeling sick. So in the case of COVID, once people get really sick, they stop going out and interacting with people. And so the number of people they infect once they have symptoms is relatively few. The problem is that you can have a high viral load and not be symptomatic. And during that period, you can infect quite a few people. And so this time scale mu for the duration of the period over which you're infectious is going to be a property of the virus, the host immune response, and also people's behavior. On the other hand, this is a number that is bandied about a lot. The key thing to remember is that it's not a property of the virus in isolation. It's a property of the virus in the context of social behavior. One result of that fact that it's context dependent is that estimates of R naught vary a lot for the same disease. On the other hand, you could get rough numbers. And if you look this up, for example, in Wikipedia, you'll find that the numbers that they give for R naught for the same viruses are quite different from the ones I've got here. This one comes from the textbook. Influenza has a typical R naught of 3 to 4. Everyone who gets flu infects typically 3 or 4 other people. Smallpox had an infectivity, thank goodness smallpox is gone, but it had an infectivity somewhere between that of flu and twice that of flu. Although there are some theories that it could have been higher than that. When we get to things like measles and scarlet fever, the infectivities go up. Chickenpox was very contagious. The average person with chickenpox infected 10 other people. Measles, it's easy for an infected person to infect 20 people. And so actually looking at COVID, were rather surprisingly, given how much of a pandemic we've had, the R naught for COVID is typically estimated not to be that big. It's comparable to that for flu. Okay, so now let's take the model that we have here and extend it to include recovery. Now you've done this in homework already, but why don't you do it again here in class? We'll keep n equal to 10, gamma equals 0.1. And now let's let mu equal 1/5. The average person is sick for 5 days. If we plug in those numbers, R naught, n gamma over mu, 10 times 0.1 is 1 divided by 1/5 is 5. So that would be an R naught of 5. What happens? So I'd like everybody to try that out, okay? You can comment out if you've got all of this fancy parameter scanning. You don't need it, so you could comment that out for the moment. You'll need it in a few minutes. So is it clear what I'm asking you to do? I'm asking you to take this model that we have here for SI and make it an SIR model. And I'll give people a few minutes to try that on their own. See how people are doing. Again, if anybody needs help, please speak up. You're welcome to do my best to be helpful. Okay, I'm going to try this on my side. Again, you may very well have better ways of doing it than I do. But the first thing I need to do is add, does anybody want to show us how they did it? If somebody would like to show screen share and show your solution, that's okay. If not, I'll do it. And as we go further on in the class, I'm going to get more insistent about having you show your results. But today, it's okay if you don't want to do that. So we need to now add the diagram to show how to go from I to R. So we'll call that E2, and we'll have I goes to R, just the way we did it for the first line. And the rate at which that happens is mu times I. And this is the recovery rate. Okay, so we need to add a parameter, which is mu. And that's going to be, we said it takes 5 days, so 1/5 is the recovery rate for an individual per day. Okay. Now we'll load our model. Let's call it, it's an SIR model, so I'll rename it SIR model. We'll load our SIR model. We won't do the simulate. We'll just put this down here. We'll simulate it, say, our old-fashioned way. We'll say S is equal to m dot simulate. Zero comma 20 comma 500. And we'll plot everything, and we'll use our old m dot plot and not worry about the fancy stuff for the moment. And let me just comment everything out here for the moment. We may need this in a minute. Okay. What did I say about timescales? For this set of timescales, the epidemic takes longer than 20. So I'm going to have to adjust my simulation time to be appropriate. Let's make it 40. There we go. Were people able to get that? Are people getting something different? Again, this was a problem on the homework as well. How many people got something that looked like that? For people who had a little bit of issue, I'll show this code again. Again, I changed the name to SIR model. That could be anything. I added the one line to express I goes to R. I had to give a value for mu. I should really have given my initial number of recovereds. So if I wanted to be more formal, I'd write R equals 0, because I have no in fact. Start with no recovered people. And then I run. Did anybody get anything different from that? Everybody got something similar? Okay. All right. So one thing that's sort of interesting here is that in an SIR model, it's not always true that everybody in the population gets infected. It's possible for people to not for the epidemic to go through and leave some people uninfected. Now, with an R naught of 5, which I calculated here, the peak of the infection happens here, yellow, around 20 days, and about 40% of the population, 44% of the population is sick at one time. So for an R naught of 5, which is a little more than flu, you'd expect almost half of the people to be sick. And the epidemic sweeps through the population in about 20 days. So we have a couple of parameters that we can play with now. We could play with the initial number of people who are infected. Here, I initial. We could change the infectivity, gamma 1 and n, or we could change the recovery time, mu. One of the things that's rather interesting is what happens if you change gamma, that is the probability of infection, for each encounter that you have, and mu, the duration of the infection, at the same time. So let's try that. What happens if we reduce mu and gamma together? If we multiply gamma by 10 and mu by 10, R naught doesn't change. And I have a suggestion here, which is instead of using an R naught of 5, let's use an R naught of 2, which we get by setting gamma equal to 0.04. So let's first change gamma to 0.04 and run that and see what we get. Well, not much in this case. I started out with the infection takes too long to take over, so maybe we shouldn't do that. Let's keep it at 0.1. Okay. So what I want you to do now is I want you to multiply gamma by 10. We can do that just by multiplying by 10. And let's multiply mu by 10 too. So R naught is the same when I do this. How many people think that this is going to have the same time series? Let's have a vote. People who believe that that multiplying gamma and mu by 10 both leaves things the same. Let's have a show of hands. You know how to use the Zoom response buttons to tell it to do that. I can poll it. One person said yes. Anybody else think things are going to be the same? Define the same would be actually the time series is the same. The time of the maximum infection is the same and the fraction of people infected the same. One person said no. I'm going to wait until everybody expresses an opinion. No, okay, 2 no's. You can try it out for yourself. You don't have 3 no's. I'm still 3 people I haven't heard from. Ryan, yes. Okay. Ali and Colin, what do you say? I'm not going to go forward until you've both expressed an opinion. Don't worry, if you're wrong, the cost is 0. So no.
Sorry, I didn't have the poll up anymore. I said yes in the chat.
Okay, sorry about that. So Colin says yes, so we have 2 yeses, and Ali, you're the last one. Yes, 3 yeses. Okay, so let's see what happens. So I've multiplied gamma by 10, mu by 10. I'm going to run it. And what happens? The peak number infected is still 40%, but the infection sweeps through the population much, much faster. So R naught by itself doesn't controls the fraction infected, but doesn't control the rate of infection. What if we go the other way? What if we divide gamma by 10 and we divide mu by 10? Let me put some parentheses here so I make sure I don't divide wrong. If I do that, it slows things down. In fact, it slows things down so much that I can't even see what's going on. But if I multiply the time by 10, I get something that looks the same. So my peak height is the same, but instead of taking 15 days to get to my maximum, it's now taking 200 days. So R naught controls the amplitude of the infection, but it doesn't by itself control the time scale of the infection. So the time scale of the infection is controlled by mu, the rate of recovery. The shape of the curve is unchanged, even though the time scale changes. And so if I actually now went through and did what I just said to do here, which is change mu by divide by 10, let's just do it. Let me do it. I'm trying to think about the easiest way to do this. I could do this here. I'm going to start out with the one that we had initially. I'm going to copy this now 3 times. Not a very elegant way of doing it. And I'm going to load my model, and I'm now going to overwrite gamma 1 and mu. So in the first case, I'm going to say m dot gamma 1 divided equals n m dot mu divided equals 10. So that's the case where it's 10 times faster. Sorry about that. 10 times faster. And the other one I'm going to do where I'm going to make it 10 times slower. And now I'm going to do times equals 10, where I'm going to make everything 10 times faster. And I'm going to adjust my time scale of my axis. So this is going to assume it's going to be 10 times faster. This one's going to be 10 times slower. And let me overlay these plot. Well, let me plot them all independently first. Did I do it backwards? I probably did, didn't I? This one should have been, sorry, this is 400, this is 4. Look at my curves, they look the same. So basically, R naught controls the height of the curve, the height of the not whole number infection, the fraction infected, and mu controls the time scale. So I have 2 independent parameters. Did people get something similar to that? If I change gamma independently, let's make gamma smaller. Let's make gamma instead of 0.1, let's make it 0.04 as I suggested initially. That slows everything down. So now I'm going to have to make my timescales longer for every one of these. Maybe not by a factor of 10. Let's see. Okay, good. So if I have gamma half of what I started out with, 0.04 instead of 0.1, it's about a half of that. What I see is that my total fraction infected maxes out at about 20% of my population, and about 20% of my population never get infected. That's for an R naught of about 2. Okay, so now I'd like you to play a little bit with this. Why don't you try changing gamma, which represents the infectivity of an infected individual per encounter. And let's try a bunch of different values of gamma and figure out what the values of R naught are for each one of those. Remember, R naught is n gamma over mu, and mu was set to be 1/5. So if gamma is 0.1 and n is 10, R naught is 5. As I make gamma smaller, the total number of people infected will go down. As I make gamma bigger, total number of infected people will go up. So why don't you try by hand, or if you want to write a little parameter scan, you can do that, seeing what happens as I change my values of gamma. So let me do that just to get started. Let's try gamma. Here we have 0.04. Let's try 0.01. If I make it 0.01, nothing happens. What if I make it 0.02? Still nothing happens. What if I make it 0.5? 0.05, I said 0.5. 0.5. In this case, the infection happens very quickly. Something that you might want to do, just so you have it to hand, is to print R naught. So why don't we print, we have to load the model first. So let's print R naught. Print R naught equals, and how did we define R naught? R naught was m dot n times m dot gamma 1 divided by m dot mu. So this is an R naught of 25. So gamma equal 0.5 gives us an R naught of 25. Gamma equals 0.1 gives us an R naught of 5. Gamma equals 0.04 gives us an R naught of 2. So why don't people try changing these numbers and see what happens? And I've got a comment here that you may have to change the time scale for the simulation to get something that makes sense. The interesting things are going to happen when R naught gets to be small. .04. What happens if R naught is, what if I make gamma equal to 0.02? What's going to happen? Anybody want to guess? Do I have an epidemic if I set gamma 1 equal to 0.02? Yes or no? Somebody, you can put it in the chat if you don't want to use the buttons. Gamma equals 0.02 is R naught equals 1. Do I have epidemic? I'll wait till people answer. Try it out. Is it clear what the question is? People should feel free to unmute and ask if it's not clear, or ask in the chat. Let me do 0.05. 0.05. Most of the people in the population get sick. R naught is 2.5. So what happens if I make gamma equal to 0.02?
What is our parameter for an epidemic?
Sorry, say that again. I didn't.
So the question is, do I have an epidemic? What is the what defines an epidemic for this analysis?
Well, to do a large fraction of the population gets sick, I would say is the definition of an epidemic. If nobody gets sick, there's no epidemic. If everybody gets sick, you definitely had an epidemic. So if I look again, if I started out with R naught, if I start out with gamma equal 1, I have R naught equal 5. Essentially, everybody in the population gets sick. And they get sick pretty fast. If I set gamma 1 equal to 0.05, that's R naught 2.5. 90% of the population gets sick. So those are definitely epidemics. If I set R naught equal to 0.04, that's R naught of 2, sorry, gamma equal to 0.04, that's an R naught of 2, I still get 80% of the population gets sick. And so my question is, if I now set gamma equal to 0.02, what fraction of the population gets sick? So this was, since this was a homework problem, you should have the code to run this on your own. But if you don't, you can use the code we've just written together. So why don't people just type in the chat for gamma 1 equals 0.02, say 0.02. What is R naught? Somebody tell me what's R naught for gamma 1 equal 0.02. Remember, n gamma over mu. N is 10, mu is a fifth, gamma is 0.02. What is R naught? You can type your estimate of R naught in the chat. Okay, Benjamin says 1. Anybody else? Ryan, what is R naught? Mu is 1/5. Mu is 1/5. That means you stay sick for 5 days, or you stay infectious for 5 days. Ryan says 50. Anybody else? N is 10 times 0.02 is 0.2 divided by 1/5, which is multiplying by 5. 0.2 times 5 is 1. So this is R naught is 1. Good. When I have R naught equal 1, what fraction of the population gets sick during the outbreak of the disease? You can try it yourself if you like. Just run it, see what you get. Even if you don't want to run it, you could think about what you've heard in the news. Why is it important to get R naught less than 1? What is supposed to happen when R naught is less than 1? Right, somebody who, okay, so that was Ryan says, when R naught's less than 1, you get geometric progression that's diminishing. Okay. So in this case, we're starting with only one infected individual. So if R naught is less than 1, how many people will get infected by that individual? It's not meant to be a trick question. Maybe it's too simple, and so it seems like a trick question. A fraction of a person, less than 1. So if you start with one infected person, he infects less than 1 person, and that less than 1 person infects less than 1 person, and she infects less than 1 person. The total number of people who are going to be infected is going to be very small. So let's actually run it. Essentially, nobody gets infected. So if R naught is less than 1, we don't get an epidemic. Even if we start out with a lot of infected, suppose we start out with a lot of infected individuals. Suppose we start out with 1e⁵. So 10,000 infected individuals. Sorry, 100,000 infected individuals. What do we see? If we start out with 100,000 infected individuals, the number of infected individuals goes down very rapidly. And basically, we get a few new infections. We do get a few new infections, but not very many. And so this is the reason people are so focused on R naught. If you can push, if R naught's bigger than 1, the number of infected people goes up exponentially. And then it peaks and goes down again. If R naught is less than 1, the number of infected individuals decreases exponentially. So the disease disappears from the population. So now you could say, as a function of R naught, What's the fraction of individuals that become infected? When does the number of infected individuals peak? And what's the maximum number of infected individuals? And here I've done a scan of this. I'm changing gamma. And here I start out with R naught equal to 1. And the infection doesn't take off at all. I effectively have no. Let me go back to the case where I start with one infected individual. If R naught is 1, there's no epidemic. If R naught is even a little bit bigger than 1, make it 10% bigger than 1. The time scale is slow, but I'm still getting an infection, an epidemic. So let me increase my time scale so I can see what's going on. People should do this with me. Oh, we're doing it. Okay. R naught is 1.09. And you'll notice that about 10% of my population, say 20% here, 20% of my population gets infected. So there's a huge difference between an R naught of 1 and an R naught of 1.01. Big difference, or 1.1. If I increase it even a little bit more, now I have 35% of my population infected. If I make R naught 2, I get 80% of my population infected. So between R naught of 1, when none of my population gets infected, and R naught of 2, when 80% of my population gets infected, that's a huge difference for a small change in R naught. And that's why controlling the infectivity of individuals by using something like a mask is so important. If a person wears a mask with COVID and they're infectious, their probability of infecting other people goes down by a factor of 2, maybe even a little more than that. That drops R naught by a factor of 2. If you can move R naught from 4 to 2, you don't do much because the fraction infected goes from 95% to 80%. But if you had an R naught of 2 and you bring it down to 1, the fraction of the population who are going to get sick goes from 80% to 0. And so reducing the infectivity of an infected individual by a factor of 2 can make a huge difference. If I'm wearing a mask and you cough on me, it also reduces my probability of infection by a factor of 2. So if both sides are wearing masks and you reduce the probability of infection by 4 per encounter, and you started with any R naught that's below 4, you've eliminated the disease in the population over a period of a few weeks. And that's why mask wearing is so effective if it's done properly at eliminating epidemics, at least respiratory epidemics. Again, near R naught equals 1, very small changes in R naught have very big effects on the total number of people infected. We can actually do this analytically. So if we look early in the epidemic, when the infection is just getting started, essentially everyone is susceptible. So S over S naught is about 1. And so that means S over S naught disappears. The rate of new infections is just N gamma times I. And so now I can write my ODEs. di by dt is R naught S over S naught minus 1 mu I. And so early in the infection, di by dt S over S naught's 1, I just get R naught minus 1 times mu I. So I get the growth rate of my infection is approximately R naught minus 1 mu. I don't need to remember many differential equations, but the exponential is one I should remember. di by dt is equal to R naught minus 1 mu I. The solution to that is I goes like e to the R naught minus 1 mu t. So the exponential growth rate is R naught minus 1 times mu. If I remember what my definition of R naught was, n gamma over mu, I get n gamma minus mu. This first form may seem a little paradoxical because it looks like the faster you recover, the more serious the epidemic is. But that's because mu is hiding in R naught. So the exponential growth rate of the infection is a function, is proportional to R naught minus 1. So around 1, if you're just a little bit above 1, you have exponential growth. If you're a little bit below 1, you have exponential decay. Alternatively, you can look at it this way. It's n gamma minus mu, the number of encounters per day times the infectivity per encounter minus the rate of recovery. In this case, it's clear that if people recover faster, you have fewer infections. If people encounter more people, you have more infections. If the infectivity per encounter is higher, you have more infections. Okay. If I look at the rate of new infections, the rate of new infections is just R naught times S over S naught mu I. S over S naught cancels. And so that means that the rate of new infections is just R naught mu, which is n gamma. So very early in the course of the disease, people haven't had time to recover. And so I can't see the value of mu. Let's think about the end of an infection. Remember, we saw that if we looked at the rate of new infections, it went up exponentially rapidly and then decayed slowly. There was a slow tail. When the infection is dying out, almost everybody's recovered. S over S naught is nearly 0. And then the rate of new infections is approximately minus 1 times mu times I, or e to the minus mu t. So the increase of my rate of infection early on in the epidemic gives me a measure of mu times gamma. That clearance rate at the tail of the epidemic gives me an estimate of mu. And so if I look at the upside, I can get an estimate of mu gamma. I'm sorry, mu n gamma. If I look at the tail, I can get an estimate of mu. Let's see, looking at the time, I don't think we're going to do this today. One thing that you're going to find in the literature, and this we'll do next week, we'll come back to the earlier problem and do that together to round out the class. But for today, I'll just say, if we look on a site like Worldometer, it'll give you an active number of active cases. And that is I in principle. I is the number of active cases, the number of people who are actively infected at a given time. And you would say, okay, that's great. I fit the beginning of this curve and get n gamma. I fit the tail of this curve and get mu. And that would be true if the number of active cases were something that was actually measured. But if you read the fine print in the epidemiological data sets, you'll find out that the number of active cases is actually a guess. And the way they guess it is precisely the inverse of what we've done here. They look at the number of cases that are measured, new cases, because reported new cases are a real thing. Number of people going into the hospital, the number of new positive COVID tests. Those are real. But nobody really checks on how long it takes you to recover. If you're in the hospital and you're discharged, then they might say the case is no longer active. But if you have a positive COVID test, there isn't any way of checking to see, unless you have tests every day, and only famous people and politicians do that. There's no way of finding out when you're done with COVID. And those numbers aren't logged. So in fact, they assume that people recover at a given rate. And so actually the tail of this curve is based on an assumption that COVID lasts 2 to 3 weeks, which is mu is 0.05 to 0.07 per day. And so this caught me last year because I saw this beautiful active case curve. I said, we fit N gamma with the leading edge. We fit mu to the tailing edge. Lo and behold, we got a value for mu of 1/3 weeks. And we said, well, that's great. But in fact, the reason we get the tail the way we do is because the data set was artificially created. Okay, let's look at the comments here. Brian says, ideally R naught should change over the course of the pandemic because of factors like immunization and herd immunity. And yes, if you add vaccinated people, that takes people directly from S to R without going through I. Good point. Maybe we should talk about that precisely. So as the epidemic progresses, let me come up with a better value of mu that's going to show us better what's going on. As the epidemic progresses, the number of uninfected individuals goes down, S goes down. Remember that the number of new infections depended on S. And so the epidemic will spread if N gamma S over S naught minus mu is positive. And I can rearrange things to say that that's equivalent to saying that R naught is greater than S naught over S. As the epidemic progresses, S goes down, and so S naught over S goes up. So the disease will stop spreading when the number of susceptibles is low enough that S naught over S is bigger than R naught, or I can rearrange that and say that when the epidemic will stop when S over S naught is less than 1 over R naught. So if R naught initially was 2, the epidemic will stop when half of the population is infected. If R naught was 5, the epidemic will stop when 80% of the population is resistant. And that's the concept of herd immunity. You can either try to change R naught, the base R naught, or you can try to reduce the number of susceptibles appropriately. And this is another reason why R naught is important. If R naught is 2, I have to either have half the population get sick or vaccinate half the population to stop the epidemic. If R naught is 4, then I have to vaccinate or have infected 75% of the population. If R naught is 5, then I have to vaccinate or have infected 80% of the population. And so as R naught goes up, it gets harder and harder to reach herd immunity. I don't think I'll jump to that one for today. So things get more complicated if the immunity doesn't last forever, because then you have an even higher threshold because you keep turning your R's back into S's. And if you do that, then you find that you have to get an even bigger fraction of the population vaccinated. But a reasonable guess for the base R naught for COVID with moderate to low social distancing is that R naught is 4 to 5. And that would mean that in order to get herd immunity, you need 80% or more of the population to be resistant, either through vaccination or having gotten the disease. As we know, in the United States, the fraction vaccinated is about 60%. And the fraction who've had the disease and recovered from it and are probably immune is on the order of 5 or 10%. And so we're way, way below the threshold for herd immunity, which is why we're seeing this big outbreak now. It also seems, of course, that the Delta variant is more infectious and that the resistance given by the vaccine is lower. But one of the problems if you have an R naught, intrinsic R naught of 5 or 6, is that the fraction of the population in the United States that's under the age of 12 is pretty substantial. And so just the number of children who are unvaccinated by itself makes it almost impossible to get to a threshold of vaccination that gives you herd immunity. You'd essentially have to vaccinate 100% of the adult population to get to herd immunity. And so that's a real issue. One of the fundamental results of this kind of analysis is you have to vaccinate children if you want to get rid of the disease. Okay, so we're basically, I think we're not going to have time to go into more detail now. I hope I appreciate people's patience with the technical snafus earlier today. Having said that Zoom would work fine this way, I guess I was tempting fate to have Zoom fail on me. So I appreciate your patience on that. Are there any questions about this issue of herd immunity and R naught? Because these are pretty fundamental concepts, and they're concepts that affect our lives every day. When you talk about vaccination strategies, when you talk about whether you need social distancing or not, whether you talk about whether you need masking or not, these are all fundamental questions that affect how public policy is derived and how many people are going to get sick and how many people are going to die. So for next week, I would like people primarily to focus on doing the thinking about your project and working on that, trying to hook up with each other to get project proposals. People gave sort of version -1 versions of project proposals today. For next week, try to keep developing those a little bit. Do try to meet with me or Giuliano or Josh or some combination of us together to discuss what's a reasonable project proposal. Because as I mentioned, some of the projects that were suggested were really neat, but would be a little bit outside the scope of this course. And I won't say absolutely no to doing those, but I think you're making your life a little harder by trying to do those. I will ask you as a homework problem to do this exercise 3.3, which would be to plot as a function of gamma and R naught, the fraction of individuals who get infected, when the number of infected individuals peaks, and the maximum number of infected individuals at any given time. You can use the code that we worked together in class. It shouldn't be too time consuming, so you still have most of your time to work on the project. But I'd like people, I know people were having some issues following along and doing the exercises today. And so it's important that the quiet of your home, when you have the time, you're not under pressure in the class, I think it's important to try to practice those things a little bit. So I will assign this exercise 3.3 as a homework assignment. It may take me a day to get that typed up. And I'm also maybe going to do one other small assignment related to this. But I think that should be, I hope that working with this will be useful for you. Almost everything else you do in epidemiology is a simple generalization of SIR models. You have SIRS models where the susceptibles, the resistant people lose resistance and go back to being susceptible. You have SEIR models where people are not infectious for a few days after they were infected. You have SIRD models where some fraction of the people who get infected die. You have models where you have new births. Those are typically for slower diseases than this one. So you have always more uninfected individuals joining the population. You have vaccination models where the vaccine, as Giuliano said, takes you from S to R without going through I. All of these models have the same basic structure, although you can build more and more complex networks.